now I'm going to welcome Professor Junke ZHENG. So are you here? Yes. So Professor Junke ZHENG is professor and principal investigator at Shanghai Tao Tong University College of Basic Medical Science, and is focusing on the regulation of stemness of hematopoietic stem cell, and to understand how bone marrow niche control the cell fate of these hematopoietic stem cells and leukemia initiating cells, and in order to dissect the potential niche components that are critical for the maintenance of this cell activity. So thank you, Dr. Junke ZHENG, to be here, and it's a pleasure to give you the word.
Okay, yeah, can you see my slide?
Yeah.
Yeah, okay, yes, thanks for the nice introduction. So it's, I think it's a good morning or bonjour in France, and good evening in China. So dear friends, so it's my great honor to have this opportunity to talk about some work related to the normal functions of immune inhibitory receptor in Leukemogenesis. So as mentioned by another speaker, I think a while ago, we know that the hematopoietic stem cells can give rise to all the progeny cells and all the differentiated cells, including B cell, T cell, and NK cells. So it's also helping consider that hematopoietic stem cells or progenitors may easily accumulate many genetic mutations, which may lead to the transformation of the leukemia stem cells, which can undergo clonal expansion to give rise to the leukemia development. So, but how to treat leukemia is still challenging. So there's, especially for some type of leukemia, as I will mention in today's topic, such as acute myeloid leukemia. So because, currently the radiotherapy and chemotherapy has been commonly used for the leukemia treatment, although it seems there is some good effect, but relapse rate is still high. One of the reasons is maybe as mentioned by Dr. Dunn, so there's some immune response are involved in this process. But another challenge is also is about the current treatments is lack of very ideal molecular targets for different types of leukemias. And secondly, also people believe that hematopoietic stem cell transplantation has been considered is a very effective and a curable way for the leukemia treatment. However, how to get enough hematopoietic stem cell for the transplantation is still challenging. So it's there's a lot of people try to find a different way to get enough amounts hematopoietic stem cells, for example, through the in vitro culture or in vitro expansion, but still how to expand the hematopoietic stem cell in vitro is still face a lot of problems. So the good thing is recently, I think also I mentioned by some other speakers, so immunotherapy has maybe a potent way for the leukemia treatment or cancer treatment. So because there's some immune checkpoints, including the PD-1 and CTLA-4 has been found by the famous scientists Tatsuki Hojun and James Ellison. So it seems very effective for treating some solid cancer, but for leukemia treatment, it seems the immune checkpoint still has some difficulty. However, the CAR-T therapy has been found it's very effective in one type of the leukemia. It's called a B-cell acute lymphoid leukemias. But for the AML leukemia or to how to target the AML leukemic stem cell, it's still difficult. Although people have found that in the leukemia stem cells, they also express many surface markers as shown in this slides, including the PD-L1 or TIM3 and CD47 or CD96. This marks it looks like the potential target for the antibody-based immunotherapy or CAR-T-based therapy. But currently, the difficulty is how to find the potent molecular targets for the leukemia stem cells. So we have worked on one of the protein is called angiopoietin-like protein on the leukemia stem cells. And our previous studies show that the angiopoietin protein can bind to one of the immune inhibitor receptor. It's called AOIRB2, so which is, stands for the leukocyte immune globin-like receptor. So they can, although the AOIRB2 has been found expressed on many myeloid cells to inhibit the immune response, but to the leukemia stem cells, it's exactly to enhance the proliferation of leukemia stem cells. And then later on, we also find that the angiopoietin protein can exist in the exons. Interestingly, also we found that this protein also can bind to the AOIB2 receptor to enhance the self-renewal leukemia stem cells. So this result give us very interesting insights. So we think that except to the AOIB2, maybe there's some other type of the immune inhibitor receptor also co-sustains leukemia stem cells activities. To answer these questions, so we performed an antibody array and to try to identify another immune inhibitor receptors. Indeed, we find some other type of candidates such as CD244 and some other members of LRBP also expressed on leukemia stem cells. And we also find maybe they also very important for the leukemogenesis. For example, we found that the CD244, which initially found mainly expressed in the NK cells, interestingly, they also can express the leukemia stem cells and co-interact with the c-KIT to enhance the recruitment of the SHP2 to dephosphorate the P27 to increase their stability to and eventually to enhance the self-renewal of leukemia stem cells. So today I'm going to talk a little more about another molecule. It's one of the AOIB family. It's called AOIB4. As I mentioned a while ago, AOIB4 is similar to the AOIB2. So this type of the immune inhibitor receptor mainly contains several ITIN domains. So interestingly, we found that AOIB4, the ligand is still it is still unknown, and also in the mouse, its mouse homologue is called GP49B1, and basically AOIB4 also is mainly found expressed on many myeloid cells, including monocytes, macrophage, and also is adversely regulates the immune response, but its function in stem cell and remains largely unknown. So we are quite interested in these molecules, and first we examine its expression on different types of acute myeloid leukemia. It's called AML, and interestingly, the AOIB4 mainly expresses the M4, M5 subtype of AML cells, and its expression level is much higher than the normal monocytic cells, and more importantly, AOIB4 is adversely correlated with the overall survival of AML patients. Then we knock out the AOIB4 in one of the AML cell line. It's called THP1 cells, and found that this knockout can dramatically delay the cell growth and in vivo, and also dramatically extend the overall survival in the recipient mice. And we also found that the knockdown LB4 can inhibit the leukemia cell migration either by assay, by a transfer assay, also can by an in vivo assay when we transplant the knockout cells into not skilled recipients. We found the infiltration in different organs, including bone marrow, liver, and spleen, is dramatically decreased after 1 day or 21 days transplantation. So we also overexpress the LIB4 in another murine cell line, AM cell line. It's called C1498 cells. And consistently, we also found that overexpression of LIB4 can enhance the cell migration and increase the infiltration in different tissues, including liver, spleen, and bone marrow. And more interestingly, we also find that LIB4 may can suppress the T cell proliferation, as shown in here. So when we co-culture the T cells with the LIB4 knockout THP1 cells, we found that cell cycle is dramatically changed. They can increase the number of the S phase in T cells, so indicating they can enhance the T cell proliferation after LIB4 knockdown. So also we found that LIB4 can enhance the T cell activity, because when we in this co-culture system, we found that after co-culture with the LIB4 knockout THP1 cells, some T cell exhausted marker such as PD-1 is dramatically decreased, which is consistent with the increased cytostatic effect when as targeted to the THP1 cells. So to further confirm this, so we performed a rescue experiment. So we overexpressed both wild type or intracellular domain defect mutant in the LIB4 knockout THP1 cells. In that case, we found that indeed overexpression of the wild type LIB4 can rescue the phenotype of the co-culture with the LIB4 knockout THP1 cells, but not the intracellular domain defect ones. So both in the increased, both as evidenced by the infiltration in the bone marrow or liver cells or the T cells numbers. So that indicates that LIB4 intracellular signaling is very important for the leukemia cell infiltration and the T cell suppression. So to confirm if this target has the potential effect in the leukemia treatment, so we eventually generate the LIB4 functional blocking antibody and to test its effect in the leukemia treatment. So as shown here, we found the injection with one injection with the functional LIB4 antibody can effectively inhibit the leukemia development in the recipient mice, as revealed by this luminescence image, and which can further dramatically increase the overall survival of the recipient mice. And furthermore, so we wanted to test if this antibody also work on the human primary AM cells. To do that, we also treat the transplanted AML cells in the not skilled mice, and then followed by the treatments with the functional antibody LIB4, as shown here. In different samples, we also can see that functional antibody can effectively inhibit the leukemia development. And also the antibody can inhibit the primary cell infiltration in the bone marrow, liver, and the spleen. Also, we found that this antibody can suppress the T cell activity in vitro. So as showing here, so when we co-culture the CD33 positive AOIB4 positive, but not AOIB4 negative cells, they can effectively affect the T cell expression, T cell proliferation, both from the normal patients or normal human, control human or AML patients. And we also indeed found that the functional antibody can enhance the T cell cytotoxic effects. So as shown here, so after treatment, they can dramatically increase the CD3 positive cells in the mice and decrease the GF positive leukemia cells, which is consistent with increase the many cytokines secreted by the T cells, including interleukin gamma. So to further understand how AOIB4 can mediate the signaling in cell infiltration and T cell suppression, so we then try to screen and identify the ligand for the AOIB4. So in this experiment, we use the chimeric report system. So as shown here, so basically we fused with the extracellular domain of the AOIB4 with the intracellular domain of the PIAOR, which can further recruit the DAP12 to enhance the NFAT pathway to drive the GFP expression. So eventually we find that both human serum and mouse serum can enhance the reporter cells to express the GFP. So indicating there's some ligand exists in the serum. So then we further use the mass spectrometry. Eventually we identified such one of the components is called ApoE, serve as the ligand of the AOIB4. So as shown here, we found that ApoE can dramatically enhance the reporter cell to express the GFP. And when we cultured with ApoE knockout serum, so in that case, the GFP expression is dramatically decreased. So this indicating indeed the ApoE serve as the ligand of AOIB4, and we also can directly use the recombinant ApoE to bind the receptor of AOIB4 by using effects analysis. And also we found that when we knock out ApoE in THP1 cells, they can also dramatically increase the T cells in the in vitro culture system. So later on, we also want to ask how AOIB4 mediates signaling controls the M cells migration and the T cell suppression. So because as I mentioned, so basically AOIB families, they can recruit the SHIP1 or SHIP2 to mediate the downstream signaling. And in that case, so we examine the SHIP1, SHIP2 phosphorylation levels in AOIB4 knockout THP1 cells. So as is shown here, we found only phospho-SHIP2 is dramatically decreased, but not the phospho-SHIP1 or SHIP. And if we knock out the SHIP2 in the THP1 cells, we can reverse the effect of the in the increase of the CD3 positive cells in the in vitro culture system. And also we can reverse the infiltration in the tissues, including bone marrow, liver, and the spleen. So that means the SHP2 serve as the main downstream targets of LRB4 mediated signaling. And then we also wanted to further know what's the downstream of the SHP2, and eventually we performed the RNA sequence in Y-type and LRB4 knockout THP1 cells. In that case, eventually we identified that the JAK stage pathway and MF-kappa-B pathway is dramatically decreased. Then we further confirmed by the Western blots, and as shown here, we found that phosphor IKK alpha and beta is dramatically increased upon the knockout of the LRB4 in THP1 cells. And interestingly, we also found that the nuclear form of the NF-kappa-B is dramatically decreased in the LRB4 knockout THP1 cells, indicating NF-kappa-B may play a role in the downstream signaling. And indeed, that when we use NF-kappa-B inhibitor, it's called B11 782. In that case, they can dramatically increase the CD3 positive T cells in Y-type, but not LRB knockout THP1 cells in the co-culture system. So then we also want to further to know what's the downstream target of the NF-kappa-B in these pathways. So we performed another in vitro culture experiments. So we culture with the both Y-type and the LRB4 knockout condition medium with T cells without contacting. So in that case, we also found that knockout condition medium can dramatically increase the CD3 positive T cells proliferation, indicating there's something existed in the supernatant. So eventually we identified that the UPA basically is the urokinase receptor, and also arginase 1 is play a role in the T cell suppression. So as shown here, so when we add the UPA and also arginase 1, they can dramatically decrease the T cell proliferation, both in Y-type and LRB4 KO THP1 cells in this co-culture system. Also, again, arginase 1 is also have similar effects in inhibits the CD3 positive T cells, both in LRB knockout, ApoE knockout, and SHP2 knockout. So this indicates they serve as the downstream target of the NF-kappa-B. And if we overexpress the UPA,
I'm sorry. Yeah. You should think about maybe shorten your talk. I don't know if you.
Almost on the last slide. Oh, thank you. Yeah. Okay, so when overexpressed, they can also reverse the effects of in the infiltration of the tissues. So basically, we eventually identify that ApoE, they can bind to LRB4 to further recruit SHP2 to enhance the expression of the NF-kappa-B expression, which can further and enhance the expression of UPA and arginase 1 to exert the T cell suppression and enhance the infiltration of the leukemia cells. So with that, I will end my talk with the acknowledgement of all my lab members and also collaborators. Thanks.
Thank you very much. So thank you very much for this talk. And so if anybody in the audience have a question? Nobody? Maybe just one. I'm very impressed by the efficiency of your antibody. Mm-hmm. And of course, when we are using antibody as a therapeutic agent, we are still thinking about side effect. So How do you manage actually?
Yeah, this is a very good question. So yeah, currently it's maybe difficult to avoid any side effect, but I think our good thing is our antibody maybe is neutralized antibody. So it's since there's little effect in some other effect in such as ADCC or so. So it's maybe dependent on the different antibody to try to avoid the side effect. Also, we can try to I think to optimize the affinity or some other issues such as to modify the FC the compartment to to reduce the side effect. But I really think it's maybe take time to to reduce the side effect. So it's still there's a lot of things we had to do to reduce the side